I'm Alessia David, and I'm going to present the Missense3D portal, which is a suite of algorithms for the prediction and interpretation of missense variants using 3D structural data only. So we know that the availability of 3D coordinates of proteins alone or in combination with other molecules allow us to calculate all those physical and chemical bonds that are crucial for the correct folding end function of a protein. And the availability of the 3D coordinates is also very important because it allows us to do an atom-based analysis of missense variants. Over the last few years, and especially with the development of the very latest modeling tools like AlphaFold, we have seen a substantial increase in the coverage of the proteome for humans, but also for other organisms. When we think about human, almost 75% of human proteins have some sort of structural coverage. And when we look at residue level for the human proteome, we have structural coverage for almost 60% or slightly more than 60% of the human residues. When we think about the impact of an amino acid variant, we consider all the possible mechanisms by which an amino acid change can affect the function of a protein. We need to consider not just the impact on the single protein, but also the impact that this amino acid change can have on the interactome. We know that proteins don't work on their own. They work in connection with other molecules, other proteins or DNA or ligands. And we and others have shown over the years that variants that occur on the surface of the protein, and especially in these surface regions that contribute to the protein-protein interaction, these variants are very important cause of disease, especially in humans, but also in other organisms. Currently, the structural coverage of the interactome in human and other organisms is not the same as with the single proteins, but this is likely going to change in the near future, thanks to the very newest algorithms that are available now. And we have the AlphaFold multimer, the very latest release AlphaFold 3. And these algorithms have already been used by groups to perform large-scale analysis of protein complexes, and models for homodimers, heterodimers are already available. So the structural coverage of the interactome is going to increase substantially. We can predict that in the near future. So in 2019, we released the Missense3D algorithm. This is for globular proteins. And over the years, we have expanded the portal, and it now includes a Missense3D for variants at protein-protein interface, and a Missense3D to analyze the impact of proteins that occur within transmembrane regions. And I'm going to talk about these predictors briefly in the next few minutes. So at the core of the Missense3D algorithm is the generation of a mutant structure from the wild-type structure. So the way in which this automated pipeline works, it takes the wild-type structure, identifies the wild-type residue that harbors the variant. It removes the side chain of the wild-type residue. It removes the side chain of all the residues within a 5 angstrom radius, and then introduces the mutant side chain and repacks the structure. So now we have a wild-type and a mutant structure, and the pipeline automatically performs an atom-based analysis of the two structures. And if any of these 16 structural features that are presented on the right are identified, then the variant is predicted damaging. And the structural features are disruption of disulfide bonds, salt bridges, hydrogen bonds, introduction or removal of charges, especially within the very buried core of the protein, clashes, formation of cavities, and so on. On our test set made of variants that from ClinVar that are known to be either benign or pathogenic. We showed that each of the 16 feature is able to distinguish between a benign and a damaging variant. But we wanted the Missense3D algorithm to work not only on experimental structures, but also on models that, as we have seen, form the majority of the coverage of the proteome. So what we did, we used our in-house homology modeling tool, FIRE2, and for each experimental structure, the test set, we generated models using templates with different sequence identity. And what we showed is that the results obtained with Missense3D on this set of variant, benign and damaging variant, the results are similar if we are using an experimental structure or if we are using a model generated with either a very good template with a very high sequence identity at the very far end on the right of this slide, but also if we use models that are generated with templates that have even a 30% sequence identity. When it comes to variants that are in transmembrane regions, we know that the lipid bilayer, the characteristics of the lipid bilayers are different compared to the aqueous environment. And this is the reason why we developed a tailored algorithm, the Missense3D TM, for transmembrane regions. This automated pipeline, the first step in this automated pipeline is to insert the query protein that has the variant of interest within the lipid bilayer. And we do this by using an in-house software that places the protein within the standard lipid bilayer and then selects the orientation that has the minimum energy. In this way, we identify, using this structural method, we identify the residues that are transmembrane. And of course, this is a structural method. It's not sequence-based. So there might be some very minor discrepancies if we look at residues that are predicted transmembrane using a sequence-based method. Once we have identified and placed the protein within the transmembrane region, the algorithm automatically performs the in silico mutagenesis, generates the mutant structure, and then, similar to what we have seen before, analyzes the wild type and the mutant, and looks at disruption of the 16 structural features that we saw before, but also these additional features that are highlighted with the yellow bullet. If these features are identified in a transmembrane residues that is exposed to the lipid bilayer, then the variant is considered damaging. For variants in protein-protein interaction site, we have developed the Missense3D PPI for protein-protein interactions. In this case, the 3D coordinates that are required for the algorithms are those of a complex, so at least 2 chains needs to be present in the 3D coordinate file. Similar to what described before, we perform automatically an in silico mutagenesis. This time, the area, the environment, is around the 2 chains, so is the variant that is at an interface, and the interface is automatically calculated by the program. All the features that are highlighted here with the yellow bullet are studied in the mutant and wild type structure, and if any of these is identified, the variant is predicted to be damaging. For variant at protein-protein interaction site, we also look at formation, not just disruption, but also formation of salt bridges or H bonds. So all those links that can stabilize an interaction between 2 proteins when it shouldn't be. And we also look at the disruption of glycine, threonine, and tryptophan residues because these residues are enriched at interface. So disruption and substitution of these residues is also considered a damaging feature. We can see here that on the benchmarking, the Missense3D programs perform equally well or better compared to other programs, similar programs that use just structures to predict the effect of variance. But I also want to show you some results on unseen variants. These are the results from the collaboration with the laboratory of Dr. Ben Jones in the Department of Medicine at Imperial. And we studied a number of variants in a GPCR protein. And we can show, we show in this figure that there is a very good concordance between the predictions from the Missense3D algorithm and the functional effect that was studied in vitro. In gray are the variants that are predicted damaging by Missense3D, and they are all below this threshold of 1. So it means that they either, in the in vitro experiments, they either the protein failed to be expressed on the surface, or there was a functional defect. And in green are those variants that in silico were predicted benign, and no clear in vitro problem was identified. The Missense3D portal and the Missense3D algorithms have two different inputs. One is the position on protein sequence, and one is the position on 3D structure. And the reason why we have two different inputs is because we look at the position of a variant on a 3D structure. This might not correspond to what we have from the amino acid sequence reported in NCBI or UniProt. So if we choose the as input the position on protein sequence, there are some examples from the website that can be uploaded automatically. But basically, what is needed here, apart from the UniProt ID and the coordinates of the variant, is a PDB ID, so the identifier of an experimental structure. However, we also allow to use models or the user's own experimental structure. And if this is the case, for example, if the user wants to use an AlphaFold model, then it's the position on 3D structure that needs to be used. And from here, we can see highlighted in this red box, the user can upload the 3D coordinate file. This is how the program runs, very briefly. Let's say we select the position on the protein sequence and we upload all the details. This runs on the fly, and depending on how big the structure is, it can take from 1 to 3 minutes. And this is the result page. We provide a JSMall window where the wild type and the mutant structure can be visualized. And then there is also a description of the structural feature that has been identified. So any structural problem identified is displayed here at the bottom of the page in red. As I said, AlphaFold models can also be used to predict the effect of variance using the Missense3D algorithm. However, we need to remember that AlphaFold models are not 100% accurate. And as AlphaFold tells us, we need to look at this score, the PLDDT score. It tells us how well a region has been modeled. And we suggest and recommend not to use Missense3D for variant prediction for AlphaFold regions that have not been accurately modeled or are disordered. The Missense3D PPI and the Missense3D TM also have their own website that can be used. The input of the two algorithms is the same as we saw before. So there is a protein sequence input or the possibility of uploading a model. And this is the result page. So on the Missense3D PPI, the main difference is that now we can see the variant on the complex. We have the two chains displayed. And in the Missense3D TM, we have the protein embedded within the lipid bilayer. All the models, the wild type, the results can be downloaded and can also be used and visualized with other tools like Chimo or Chimera. Finally, from the Missense3D portal, we also have the catalog of over 4 million variants, pre-calculated structural predictions for 4 million human variants already available. And this is the Missense3D DB, Missense3D database. This was generated using all the known missense variants for humans. So we selected over 10 million missense variants, and we identified PDB coordinates, so experimental structures, but we also generated a large number of models using our in-house FIRE tool. And what we did, we mapped these 10 million variants onto the either experimental or model structures when an experimental structure was not available. And just to give a few numbers, we were able to map to models or experimental structures almost half of the 10 million variants, so 4 million out of the 10 million missense variants that we extracted from NOMAR, ClinVar. And we showed that 14% of variants that we mapped from NOMAD had some sort of structural problem identified. But also very interesting, when we look at results from ClinVar, 20% of the variants that are VUSs, variant of uncertain clinical significance, but also variants with conflicting interpretation, 20% at least are predicted to cause some structural damage. So these could be variants that are disease-causing, but at the moment are only predicted as VUSs because not enough information is available. The Missense3D database it's very simple to search. It requires a unique ID or a gene name. The next page will show all the variants of this. For example, is the page of the ACE2 protein. So this is the receptor for the SARS-CoV-2. And we in the database have over 249 variants, and we can search by the specific variant position, or we can select and look at all the variants for this protein that are in the catalog. So if we do the latest, so we look at all the variants in the catalog, we have this particular result page. There are several information that can be displayed, not just the structural predictions from Missense3D, but by clicking, unclicking, we could also display the ClinVar annotation or the MAF score from NOMA, the type of PDB or model that was used, the SIFT, the polyphen score, and so on. All these information can be downloaded from the page in a CSV file, but also for the specific variant, a TXT, so a text file can be downloaded and has all the information that we have in the catalog for the specific variant. And this is something that the people working in genetics find quite useful, because this is a report that can be copy-pasted, for example, into a report. But clearly, the strength of the Missense3D database is the structural predictions that we generated, pre-calculated using the Missense3D algorithm. And in the result, in the simple result page, we can already see the prediction, and also here on the far right, the type of structural damage identified, so like disulfide breakage or a cavity or a clash. But if we click on the actual prediction, so in this case, for example, damaging, we go to the specific page for the variant, where we have the wild type and the mutant structure and the variant and the wild type identified, presented. All these models can be downloaded and visualized on another, like in PyMod, for example. And we also have the structural prediction, and we have the features that have been identified, and the ones, and if anything has been identified as damaging, this is highlighted in red. As I said at the very beginning, with AlphaFold and all the new AI tools for 3D protein modeling, we now have the 2 million models for proteins, not just in humans, but also in other 47 key organisms, and these are all in the AlphaFold database. So there is quite, it's very timely. The use of the Missense3D algorithm is quite timely because we have the structures now, and the Missense3D algorithm can be applied to study variants not just in humans, but also in other species. So in conclusion, the structure-based modeling of genetic variants can aid in the interpretation of variants that are naturally occurring or also engineered. And this is quite interesting. And in some cases, it's important to know not just that the variant is damaging or benign, but also to understand why. So to have some predictive mechanisms. The recent advances in protein modeling with AlphaFold makes the structural modeling of variants, of missense variants, particularly timely, and of course offers new challenges and opportunity for the biomedical communities. And as I said, this structure-based modeling of missense variants can be performed not just in humans, but also in other species, as long as the structure is available. And with this, I would like to thank all the other members of the group, and we very welcome feedback on the use of Missense3D. Thank you.